Hey, PBio, welcome back to our last video on cellular respiration, where we talk about the final two stages. So remember last time we were talking about glycolysis in the cytoplasm and fermentation in the cytoplasm, but now we're going to be talking about those who possess mitochondria and breathe oxygen. How are they producing energy? So first, we're going to need to know a little bit about the structure of mitochondria. So you remember that it has a double membrane because it was a long time ago swallowed by our ancestor to give that second membrane. So we have an outer membrane and an inner membrane. And you'll see in this inner membrane that it is really folded up, bunched up into these folds called the cristae of the inner membrane. And because we're going to want a ton of proteins to embedded in that membrane to generate ATP in our final payoff step, um, these folds give it more surface area. So it actually becomes more efficient by having more places you can process this reaction. Inside that inner membrane is a place called the matrix, which you'll see has a home to DNA and ribosomes, because remember, this was once an independent bacteria that our ancestors swallowed for its energy producing properties. So, as pyruvate, where we last left off from glycolysis, is entering into our mitochondria, it's going to get converted into something called acetyl coenzyme A. As a part of this reaction, we're going to kick off some of our carbon dioxide, and we're also going to build another energy shuttle with NADH. Now, because we have two pyruvates, this happens twice. So, two carbon dioxides, two NADHs are made. So, two pyruvates go in, we breathe out two carbon dioxides, we make two more of these high energy shuttles that we'll be using at the end, and then we have two acetyl coenzyme A's that will have enter the citric acid cycle. So you'll notice sometimes it's shown as the Krebs cycle, sometimes it's shown as citric acid. You can use either term, they're both acceptable. And they occur in the matrix of the mitochondria. Now, this looks really complex. I don't want you to memorize every step of the citric acid cycle, but I want you to kind of see where are we gaining things. And I want you to see, just like in glycolysis, that it's a step or series of reactions that are going to take place. So here it's this cycle where oxaloacetate gains an acetyl group. And then we do a series of reactions where we're breaking down or catabolizing that product of citrate to kick off a carbon to release more CO2, to kick out some electrons, to reduce our electron shuttles. And also, some of this will release a little bit of energy as we're catabolizing to help us produce ATP. So we're going to kick off our remaining two carbon molecules from our pyruvate. So now we have no carbons left from the original sugar molecule. We've breathed them all out. We've generated 1, 2, 3 NADH molecules. We've generated an FADH2 electron shuttle, and we've made an ATP all in one turn of the cycle. But cycles are circles and they're going to keep on turning. And remember, we have two acetyl coenzyme A's for every glucose. So we're going to go through it again. That means we have 4 carbon dioxides from 2 turns, 6 NADHs from 2 turns, 2 FADH2s from 2 turns, and 2 ATP from every sugar. That's a lot of electron shuttles that we've made. And now finally, we're going to get to see what we do with them. So, oxidative phosphorylation is the last big step, and that's going to be taking place on the cristae of our inner membrane. And what we're basically going to be doing is using cell transport or building a concentration gradient and using the flow of that concentration gradient of ions to power the reaction of building ATP. So, first step is called the electron transport chain. And all those electron shuttles that we made in glycolysis, Link, and Krebs, we're finally going to get to use them. Those electrons they've been carrying have a lot of energy. They're high potential energy electrons. And they're going to flow through this complex of embedded protein channels to power them to move hydrogen from the matrix into the inner membrane space. Now you'll notice there's a lot more hydrogen in the inner membrane space than there is in the matrix. And so that requires energy. Anytime you're moving against a concentration gradient, you need to put in energy. So that's why we've been collecting all these electron shuttles. They're flowing through catalyzing a series of reactions which generates the energy to power these proton pumps. Now, if you remember transpiration, water doesn't flow out um, unless has somewhere to go. Same thing here. Electrons can't flow unless they have somewhere to go. And so at the end, we're going to actually produce water. Those electrons are going to bind to oxygen and hydrogen to generate a water molecule. So this works by the same principle as if you've ever hooked up a battery to a circuit. The negative electrons are flowing from the negative end of the battery to the positive end, and that flow is what's going to generate that electricity to, for example, power your light. Well, same thing here in the electron transport chain. These electrons are flowing through a complex of proteins, ending up in the production of water, and that flow is generating the power needed to move these hydrogen ions out of the matrix against their concentration gradient. I do not expect you to memorize all these series of reactions. I do expect you, however, to be able to explain the basics of a flow of electrons from our electron shuttles, power some proton pumps to move hydrogen ions against their concentration gradient up into that inner membrane space. So why would we do that? Well, there's a step called chemiosmosis. Also embedded in our inner membrane are these protein channels called ATP synthase. And ATP synthase is the only way for these hydrogen ions to move back with their gradient back into the matrix. So these hydrogen ions are packed in the inner membrane space and they want to spread out. They want to move back to the matrix. But because they're charged, they can't go through the nonpolar tails of the membrane. So they need a channel to go through. So what ATP synthase does is as those hydrogen ions flow through, just doing facilitated diffusion, moving without any energy needed, they're going to move through ATP synthase, which kind of acts like a rotor. It's going to spin as this flow happens, and that's going to catalyze this reaction of ADP becoming ATP. And so each hydrogen ion that goes through, we're facilitating more of this reaction. You can think of it kind of like a turbine or a water wheel. As that water or wind flows through, that's going to rotate that turbine, which is going to generate, for engineers, electricity. But our cells are pretty complex engineers and have the same basic technology. Hydrogen ions go through, that flow spins the rotor, that rotor generates the power to create ATP. So, coupled together, the electron transport chain and chemiosmosis are going to use up some oxygen to make water. It's going to use up finally all our electron shuttles, and it's going to make, like I said, the water. We've now recycled or reoxidized our electron shuttles. They can be reused back in glycolysis and Krebs, and we're going to make roughly over 30 ATP. So why don't we know how much it is? Well, unlike with substrate level phosphorylation, where it was a direct, we can see the reaction happening, chemiosmosis and electron transport chain aren't directly coupled. We're just pushing a bunch of hydrogen across with the electron transport chain, and then as they flow through ATP synthase, we generate ATP. But because it's not directly one for one, we get about 3 ATP for every NADH and about 2 ATP for every FADH2. And since we had 2 of these and 10 of these, that's at a maximum 34 ATP. But if you look at different sources, they'll say like overall in cellular respiration, we get 32 or 34 or 36 or 38 ATP. Different sources will say different things. And that's because it's not directly linked. We always get 2 from Krebs, 2 from glycolysis, and then anywhere from 28 to 34 ATP from oxidative phosphorylation. And that's why there's a range. But even at the low end, it's way more than we would get with just glycolysis and fermentation. So you can see why our ancestors swallowed a mitochondrion and kept it. So going back to our original formula, we used up sugar in glycolysis and made a tiny bit of ATP. We made carbon dioxide in the Krebs cycle and the Link cycle and got a little ATP out of it. And then in oxidative phosphorylation, that's where we used up our other reactant. We used up oxygen. We made our other product in water, and we produced a huge amount of energy in the end. You may wonder, can I burn fat? Can I burn protein? And the answer is actually yes. You can remove the amine group from an amino acid, and those proteins can enter at various stages downstream from glycolysis towards like the citric acid cycle. Fats, different parts of it as it's broken down. Glycerol will enter mid glycolysis, fatty acids will enter closer to the Krebs cycle, and these can be processed for sugar. However, it's less efficient to be processed in this cycle. And especially with like fats, there's some toxic ketone products to this breakdown reaction, which can be bad for you. So that's why we primarily use sugar, unless your body is in crisis, you're starving, in which case you're going to break down whatever you have available so that you can stay alive. That's it for cellular respiration. Catch you next time.